in Excel. It's just the easiest thing to do with this. All right. Now, just like earlier when you were creating a Manhattan plot, Manhattan plot is just a visual representation of your results from a genome-wide association analysis, right? It can be done in HaploView, it can be done in Excel, it can also be done in R and other programs. We just happen to be using HaploView. There's a variety of programs that can do it, because all it is graphing your results. As you can imagine, if you have a p-value for every SNP and you have 8,000 or 100,000 or a million SNPs, you can't really put that in the table. Right? So a graphical representation is what you get in the Manhattan plot, but also when we're looking at MDS plots, we're looking for population stratification. What might be a concern when it comes to population stratification? Well, in a case control study, if all of your cases are in one population and all of your controls are in another, you're going to have confounding effects, and you'll be not only identifying what is associated with your trait, but all the other genes that make those populations unique. Right? And I'll give you an example of that in a sec. So if you've got the Excel file open, you want to select columns D and E with the headings C1 and C2, and then choose an empty cell to the right of the data, and from the cell menu bar, you want to insert scatter plot. You know where that is on the files? While waiting on the files to open here. So highlight columns D through E. You include the headers when you do it. And then you just want to insert a scatter plot. So under the data tab, whatever, there's like a plot at the top. You can insert scatter plot based on those two columns. Now an MDS plot detects population stratification. So now we're going to learn more about this next week. However, the values in columns C1 and C2 represent the first and second principal components of variation in the SNP data. So essentially, multiple dimensional scaling measures the similarity among samples. So for example, how often they have the major allele at each locus. And it very cleverly interprets the genotyping data to show us how related individuals are in a genomic space. So as a result, individuals that are close relatives will chart near one another, while those from remote family groups will be distant. This can be used for a variety of ways. It can be used when you're looking at within families. You can look at it across breeds. You can look at it across species, depending on what the species are. Right? But the thing to remember is it is presented in a two-dimensional space. It is also possible to do this in a three-dimensional space. Right? So R has a software package that you can actually rotate it around. Because what's the concern if you're looking at it in a two-dimensional space? You guys should all see plots now. I can see some. If you're looking at it in two dimensions, it can look like two populations overlap each other. Right? But if you rotate it around into three dimensions, you can see there's actually a huge gap between them. And I don't remember what the right song is there. So for the group that's here, I don't get the demos. If you're looking at a Manhattan or a MDS plot, you have dots. Right? And we have some stars. So different populations, right? Might be hard to see the other side of the room. Stars and dots here. So it would look like maybe they're the same population. Right? But this is in two dimensions. If we look at it in the third dimension, right, what we might find as we rotate it around, these dots are here and the stars are over here. Right? But just from the view that we're taking, the perspective that we have on it, it looks like they're the same. But you want to rotate it around and look at it in multiple dimensions, and that's where we talk about principal components of two components versus three components and so on. Because it's only from one perspective. They may actually be quite far apart. Right? So think of it almost like an optical illusion. If you look like two people that they're standing right next to each other, there's no space between them, and then you walk around, and you see there's actually a huge gap between them. It was just the angle that you were looking at. And that's what happens when it comes to interpretation of MDS plots. You have to be careful. It most likely means they're fairly similar, but they may not be as similar as you think. Okay, we're going to talk more about this next week, so it's okay if you need time to think about it a bit. So the question on the slide here is, what might the clusters represent in your data? Remember, this is real world data. We know it's one breed, one type of dog. What is the dogs that you were working with? What? Kelpies. All right, and what did I mention earlier when we were talking about the background of Kelpies? We were learning more about the population. What was mentioned? Sorry, there two what? There's two distinct lineages, right? One was what? Not black and tan, nope. Lineages were what? Australian working Kelpie, and the show Kelpies, which were Australian Kelpies, right? So what you're most likely seeing here are the two different breed registries, the two different registry types that are segregating based on family history and families, right? You can see this on a small scale and a large scale. And we'll talk way more about this next week. And next week you'll see an example of where you can do it in humans, right? You can do an MDS plot of European descendants, and when you build that, you can actually rotate it around and it will just outline the countries in Europe. Right? And if you zoom in a bit more and you look at regions in certain countries, you can narrow it down even more. The more SNPs you have, the more data you have, the easier it is to find out those things that make that small population, that subset, unique. Any questions about that? Okay, so another little inspirational quote here. Success consists of going from failure to failure without the loss of enthusiasm, which I know can be challenging sometimes in genomics. Okay. So, the last thing that you're going to do today, once you've finished it, when we double check it, you are free to leave, right? Is do another genome-wide association. Now that you have experienced how association analysis is performed on a marker-to-marker basis, with a bit of guidance from us, you will now progress to running a larger analysis more on your own. The analysis that you will perform will consider mapping the genes responsible for skin color in apples. A research article by Amiet et al., which you should already be familiar with, considered the mapping of several eating quality traits for apples. Now, the study included the release of genotype data and phenotype data for red and green skin color in apples, and this is the data that you'll be using today. Now, I've already downloaded the data from the paper for you, and I've converted it to the transposed format TPET file and TFAM file. Note that the data downloaded from this project was first converted from a file that had genotypes with no spaces, extra columns, columns out of order, and a header line. The header line was used to make the TFAM file, and the identifiers for the different cultivars needed to be matched with those presented in Table 1 of the paper. Not all of the codes matched neatly, and they needed to be figured out. So this is common when using sharing data, or finding publicly available data. You should, again, never expect the data to be in the format you require for your analyses, because every analysis is a bit different, every software is a bit different, and it always takes time to do these. So when I consult, do other things with other stuff for the horses, I always tell them we need 3 months to clean the data, and I need 2 days to do the analysis. Because you'll be finding errors and mistakes, and they send you something and there's a repeat individual in there, or they send you another sample that has a different name. And a perfect example of this is there's a famous history in horses where a horse foal was born that failed the parentage testing based on all the information that was provided. Failed it, so they retested it, failed it again. The father is not the father. And then they were like, well, who is this? And they have a huge database, and they looked at it, and they found out that apparently the horse that is the sire is no longer able to have babies. He's a gelding. He was castrated very young. But at some point in his early life, he was on a property with the female horse. He jumped the fence, ran over there, had a good time, then put himself back where he belongs, and they didn't know till 4 years later, when they went to register and had issues with registration, that it wasn't the right horse. Right? Because the horse wasn't even in the same state at the time. So things happen with real data. Be aware of that when you're looking at this. So again, like I said, here's the paper in question. The files are on Canvas page. You will need to move them to your virtual machine. You can, now, we are working with apples, and how many people, I think I asked before, nobody in here is really plant, right? There are no sex for the apples, so you can just make sure the sexes are coded. Sire ID and dam ID can be coded as 0. Same thing with sex. You can code it as missing, so 0. Right? And as you know, TFAM format was clearly developed for animals, humans, rather than plants, but it still works fine for apples. For your phenotypes, red skin is 2, green skin is 1. Make sure to tell Plink to apply standard multiple testing corrections to the data. You should get a result when you do your GWAS. I mean, again, make sure you apply standard multiple test corrections to the data. And the way to check is to look and see, does your chart agree with the findings in the paper? The paper has a Manhattan plot. Does your Manhattan plot look at that like theirs? You may have also noticed earlier that the Manhattan plot that was listed here, so it's not saying dog at the top of it, it says apples. Now, as you are interpreting the data, you should be observing a substantive peak. So by this I mean there are many markers reaching significance within the general location if you've done it correctly. You may also see 2 peaks in your chart that were not highlighted by the paper. And it's because we are using a different mapping technique than what they did. They use one that accounts for population stratification. You will not be doing that yet today. So in their chart, the peaks that are seen are slightly diminished compared to what you will find. And we will get a bit more into that next week. All right? So I will leave it to you to generate the Manhattan plot. And once we say, okay, you are more than welcome to head out. We will stick around to help whoever needs help.
Sometimes I can't be bothered.
So you just image it.
Yeah, so posterior view, yeah. But like sometimes, as I did like with like the other one, I don't usually do this, but this was the week interviews and everything. So I watched flashcards so I can look at it before.
What is the order to retreat?
Oh yeah, on this.
What? Oh, what?
Oh yeah.
But it does that.
Yeah, but like, I was like more looking at this stuff because like, I kind of based this question off what the quiz kind of looks like. Because like the quiz might ask for like the function and all that. Yeah. I don't know, things like this.
These are multiple numbers. I know one of them is like the sourdough.
them all. Too much like this.
Scale on the stupid line. Is that like the...
I don't know, how would you
Okay.
I'm actually too drunk.
Oh, you mean the skeleton?
Oh, did you mean like scale up as the engine?
Yeah, I was like...
Oh no, I meant like scale up
Yeah,
as
yeah,
the
yeah.
engine. There's nothing just called.
Yeah. That makes sense.
Maybe my flashcards aren't worded to you.
I shouldn't say. I'll get used to it.
Kind of just because let's be real. Things like this. Because in this spot test, this is what it's gonna look like. Yeah, so
What's this?
it's out here.
The ear looks like I've seen it for a long time. Like, my Wonder Friend sounds really good.
Jared, can you notice? Because like this isn't this like pretty much like the quiz, like instead they'll have numbers and then for the 6 marker it'll be like a label 10 is like
Well, Boney Labyrinth. I don't know if it's called again. And then that's like the scale of... Honestly, don't know.
Oh, it is a phone. So what I do a couple rounds of these flashcards, then to pull out and then start.
After a couple of rounds, I lost all motivation.
No, you get used to it. But Anki kind of forces it all into me. Oh, you have to do it because it's December. What about the CT assessment as well? You think I can get an 80 plus?
You definitely still can. They didn't even Oh, yeah. And also,
Isn't that 30 for finals and sport test? 30, 24, 30, 20. Oh no, it's 30, 20. So it's 30. So keeping that on the hand, I think probably it's going to be like the 2 assessments of it. 30, 30, consider this an actual man. Consider it with mama is like
Yeah, like raving on them sometimes. Yeah, unless you just like fuck the data simple.
Yeah, once you filter for the results.
Sack is so dependent on the report now. It really is. 30% report comes in. What's the role?
like sensitivity, so obviously you get
You can have like a comic book face or like a Google Doc.
like you're not like putting on really old.
So we have Google Sheets. So we can put it on meetings and oh, how do you Let's do the opportunity to go for
Like trying to get rid of finding like whether Is that what she's doing right now? All the lowest songs are the lowest like you basically like improvise you like kind of probability version of the 0.05 divided by how many like tests you're It's like the main pairs or something. I guess you can just kind of keep counting them outside there.
Yeah, so what is the point of a Manhattan? Yeah, exactly. Just a visual representation of your results. Right? So even though you can't generate a Manhattan plot easily on the Tableau view right now, you can do it on R and other things with lots of programs. Right? Because you're just plotting the results, right? And you can sort your output files, and the lowest value is the most significant. So if you sort your output files based on p-value, and you find out what location that is at, and you look at the paper on Apples, they have a Manhattan plot. And you can look and see, okay, is your top marker also on the chromosome that's your top marker? If it is, then you know that's right. Second bit, which we're going to talk about next week, and also we go back. So genome-wide association is usually designed to narrow down the range. Remember, it's an array. It's an array data, and we know that those markers are not every marker, and they're just like evenly distributed across. Yeah, like it's genome-wide. We're looking at the whole genome, but we're not looking at every marker in a genome. So what happens is you narrow it down to a region, like you'll see a spike on the Manhattan plot. Like, okay, it's probably this chromosome you're missing. And then we just have to find that, and that's what we'll talk about in where we like, that's where we find the like actual nucleotide, super nucleotide that changes the protein that affects it. Yeah, sometimes it can be an insertion, sometimes it can be you guys don't do that.
Something like bit like command
much more complicated.
something like Siri as well.
If it's a Mendelian autosomal recessive, it's probably a single nucleotide. It doesn't mean that it is. There are some like Mendelian inherited, but it's a deletion. And it's just like we have two copies of it or one copy of it. You need two to have the disorder. So